Good evening, students. Am I audible? So, welcome to the fourth session, which is week four session for the NPTEL course, which is Plant Cell Bioprocessing. I'm Fenita Hepsiba, PhD scholar from IIT Madras. So today we will be. I hope that you guys have gone through the lecture videos already. So, what are the titles or what are the areas which you have seen in this lecture week, which is optimization strategies and what you have seen secondary metabolism, right? Part of secondary metabolism and part of optimization strategies. So I have few questions so that we will discuss the um, content behind that also. So I think we have waited enough. Around 5 to 7 minutes we have waited. Let's go into the questions. The question number one is: Dash are chemically diverse, broad spectrum secondary metabolites which are formed or activated only when the host plant comes in contact with the pathogen. So, what do you think the answer would be for this? What what are called the uh, secondary metabolites which are broad spectrum and formed or activated only when the host plant comes in contact with the pathogen? What would be the answer, do you think? Any answer? Phytoalexins. Yes, Ashan, you are right. So, what do you mean by phytoalexins? If you don't want to talk, you can type in the message section itself. So, what do you mean by phytoalexins? Yes, as mentioned in the question itself, these are produced only when they are when the plant is in coming in contact with any harmful organism or pathogens or some them. So, what are the other things you know about phytoalexins? These are defense metabolites. What else? These are produced even before the infection starts or only after the infection has been acquired by the plant system. Idea?
Before, I think it is before.
Okay, so you think it is before the infection starts?
At the time of the contact, when the that pathogens is contact with that the plants.
Before or after, Ashok? Whether the plant produces these phytoalexins before contact with pathogen or after contact with pathogen?
After contact.
After contact, yes. So phytoalexins are secondary metabolite compounds. These will be undetectable before infection, but they are rapidly synthesized once they are when the plants are coming in contact with any pathogen. So, have you gone through the what do you call these? They reserve their energy sources so that only when any attack happens, they will start synthesizing that particular protein which, or components which are necessary for producing this phytoalexins. The enzymatic machinery is not fully activated unless it is activated by the attack by the pathogen. So this is for phytoalexin. So um, why does the plant have to uh, cause or show some defense mechanism against pathogen? Any idea? Or I can put it like, how does the plant recognize any attack from microbial infection or herbivorous attack or something? How do they sense anything is happening to them? So, how do animals sense that we are? Yes, Sashank is right, ma'am. So, can you explain what ma'ams are, Sashank? Just brief. Can you brief it out? These MAMs are microbe assisted general molecular patterns. These we can yes, pattern recognized by receptors and plant cells. Yes, you are right, Sasha. Plants will have certain receptors which will recognize these particular patterns or components which are present in the herbivore, like microbial cell wall system or something like that. So any examples can you remember from the slides of the lecture for this particular pattern associated recognition of plant cells, like plant systems? I think this can be like, say for example, in human body also, the first line of defense and innate immunity. You can visualize that as a similar case for this particular way of detecting or coming in contact with any pathogen, infectious pathogen. So this will also be referred to as innate immunity in case of plant systems. So any example do you guys remember regarding microbe associated molecular patterns, ma'am? MAMPs. So I so in case of flagellated organisms, there is a receptor. This is the pathogen. So the certain plants have some receptor which can recognize this. There is a protein called FLS2. So plant has a receptor to recognize this particular pattern which is present in any organism which has flagella, so that it is a what that is why it is like, it can recognize a broader range. Say for example, in fungal cell wall some compound chitin is there, so if some plants might have will have actually chitin receptor, so that they can act against various ranges of fungal attacks. So that is how it works. So in case of any flagellated organism, this FLS2 receptor from the plant system can detect the molecular patterns of flagellated organism and act against most of the flagellated organism. So when we talk about plants defense mechanism, why do you think, or why do they go for defense? Like, how do they use it up further? We all know that plants produce secondary metabolites as a defense mechanism, but why do they go for defense mechanism? And also they are self-acting because they don't have any, um, what you call, some external agent or external person to come and correct them or treat them for the betterment. So most of the plants, they do have defense mechanism in the form of secondary metabolites. And have you guys gone through systemic and acquired resistance which are acquired by the plants? Let's talk about those in the further slides. So the question number 2 is, colonization of the root zone by rhizobacteria stimulates the formation of root nodules. Further, a signaling cascade involving jasmonic acid and ethylene is initiated throughout the plant. Protective measures are activated across the plant, which result in an enhanced mode of preparedness against pathogen attack. This ability of the system is termed as A. Systemic acquired resistance, or induced systemic resistance. What would be the answer? Yes, sir. So Shashank is telling that it is induced systemic resistance. So what makes you think that it is induced systemic resistance, Shashank? Uh, What is the reason behind it? So first we will discuss about induced systemic resistance, and then we will discuss about um, systemic acute resistance, which is SAR. Pathogen is not directly involved. Um, okay, so more than um, so you can put it the other way. Here, even before the pathogen's involvement, certain bacterial or fungal species. Example, for example, rhizobacteria. This is not doing any harm to the plant, but in fact, it is aiding in the formation of root nodules, like somewhat having a systemic, like a symbiotic kind of relationship. So, non-harmful organisms will induce systemic resistance so that this plant system is being entirely prepared for any further future pathogen attacks. That is what induced systemic resistance means. And what about systemic acquired resistance? What does it mean? How does it is different from your systemic acquired resistance, which is SAR? Any idea? Continuous attack of pathogen involved. Okay. So more than so some instead of telling that as continuous attack of pathogen, you can tell like how do we develop fever the same way. So after infection, how do we say for example if we got fever and the fever is due to some infectious bodies entering into our system. So after that, if the same organism is going to attack or is coming to attack us further, it has entered the body. What will happen? Our WBCs or white blood cells will be more active because they remember those particular pathogen already and have prepared for it. So the same way occurs here, where in your systemic acquired resistance, after any infection by a certain bacteria or fungal contaminants, the plant will develop increased resistance towards subsequent attacks. It will develop a preparedness due to the pathogenic attack which happened prior. So, and due to which it will send signals throughout its plant system. Like, for example, if it develops any attack in the leaf, it will generate a signaling molecule. What do you guys think about? Think that the signaling molecule in SAR in induced systemic resistance, we can tell that it is jasmonic acid and ethylene as mentioned here in the slide. So, in systemic acquired resistance, what is the signaling molecule which you think comes into picture? Salicylic acid, yes, correct. So salicylic acid will come into picture, and if in case your site of infection or site of first contact is leaf, say for example, uh, so what happens? The microorganism has entered your plant system already, and thereby some gene pathways will get activated, and this um, jasmonic acid, no, salicylic acid will get secreted and it travels throughout the entire plant system from the leaf to all the parts of the plant so that it is like a signal. It is actually a signaling molecule. So after it accumulates in the zone of infection, it travels throughout the plant system. So thereby establishing a sense of preparedness or readiness towards the attack in the other parts of the plant system. So this is your systemic acquired resistance, and like apart from salicylic acid, do you think there are some other um, signaling compounds which are involved in this systemic acquired resistance? Any idea? So uh, is the systemic acquired resistance is confined to one plant, or it is affecting the nearby plant also? So is this systemic acquired resistance is confined to one plant? Say for example, you have an entire field. So is this limited to one plant in that particular area, or it can even transfer its resistance to others nearby plants? So you guys find out in the next session that systemic acquired resistance is confined only to one single plant, or it is going to aid resistance or accelerate resistance in the, yes, maybe effect is seen in nearby plants, yes. So chance there is chance it can also affect resistance in nearby plants because salicylic acid can be somehow transferred. You guys find out what is the mechanism behind it. I hope you guys remember the pictorial representation of the systemic resistance. Let us do this as an assignment. Next time or week 5, we will discuss about this. Whether you guys went and found out about what systemic acquired resistance actually is. Thank you for answering, Sashank. So let's move to the next question. So the answer for this question is induced systemic resistance. So, what are the advantages of you having induced systemic resistance? Any idea? You guys have learned it through professor's lectures that induced systemic resistance has, of course, some advantages over systemic acquired resistance. Can you guys remember anything? Okay, we will discuss in the next class. You guys go and find out uh, the answer for this, like systemic acquired resistance and induced systemic resistance. So, in plant cell culture, cell line selection is an active process which deliberately favors only the survival of the desired variant. Is this statement true or false? Cell line selection is an active process which favors only the survival of the desired variant. True, yes, true. So, selection is favoring only the survival of the desired variant. What is screening then? Yes, Sashank, Tejas, you are right. Sashank, you are right. The answer is true. So selection is survival of the desired variant. What is screening then? Any idea? Screening of cells, cell line screening and selection. We call it like that, likewise. So what is cell line screening then? It's a passive technique. What do you mean by passive technique, Sashank? Any idea? Anybody else also? Shashank has told it is a passive technique. Okay. So what do you mean by passive technique? And other to view morphology through microscope. Okay. Okay. So you guys mean that identification of phenotype. Okay. So clumps instead of only desired variant. Okay. 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 So screening comes first or selection comes first? Screening first. Okay. Screening is first and then is the selection. Right. So we'll go to the next question. How is plant cell aggregation reduced generally to achieve a uniformly distributed fine cell suspension culture? So the options are by decreasing the oxygen concentration, by the addition of protease, by the addition of sucrose, by the addition of pectinase. What is the answer for this question? Answer is D. Okay. So the answer is D. Okay. So Shashank has given the right answer, which is if you add pectinase to your cell suspension systems, you can reduce the aggregation. So, what do you mean by cell aggregation? Or first, we will start with plant cell suspension culture. What do you mean by suspension culture and what do you mean by fine cell suspension culture? What is the difference between both? What is suspension? What is suspension culture? Fine cell suspension culture. So, what are these? Any one of you have any? Have you guys learned plant tissue culture as part of your lab? Anyone have experience in plant tissue culture? You can raise your hands if you have experience in plant tissue culture. No one of you? Okay. So then how do you understand suspension culture? What do you, like how do you single cells and small aggregates can be seen? Can be seen in what? Single cell and aggregates can be seen in culture. Yes, Shashank, the culture. So do you think it is a solid culture or liquid culture? So in culture means what? In media you have to tell, na? So culture is like cells along with your media, we can call them as culture. So yeah, liquid media. So in liquid media in conical flask you can see you can generate cell suspension culture. Okay. So what do you mean by fine cell suspensions then? So fine cells in the sense after filtering them through filter and removing aggregates. Yes. So after removing any aggregates, cells of uniform size we would say at the most. So those are called suspension cultures. So why do they even tend to form aggregates? Why they can't look similar to microbial cell systems, where they all look like you can see literally that you cannot even see the cells through your naked eye. But here you can see the cells. But why do you think that plant cells tend to form aggregates? Any reason for them in particular? Why do the plant cell systems tend to form aggregates? Plant cells communicate through plasmadesmata. Okay, they do communicate. Okay. So, have you thought about plant cell wall? Plant cell wall contains some components which holds them together. So, one of that is pectin, cellulose, hemicellulose, pectin, and others. So, they need connection between them because we are looking into um, multicellular systems, not single cell organisms like microorganisms. So here the cells need connections between each of them and they communicate also. They live together, like they are all attached together through cell wall components. So of course they will be forming aggregates. So you are externally trying to provide external force or reagents to make them into fine cell suspension. At one point, we can make them as finer cells. So, in this question, that is the answer. Why you randomly add pectinase here? Because the cells have pectin in it. So, when you add pectinase, it will cause them to disperse away from each other. So, that is the reason you are adding pectinase. So, thank you for your answer, Shashank. Let's go to the next question. So, the question 5 is Dash is a technique to enhance the accumulation of secondary metabolites in plant tissue cultures. So, given are the options. First is elicitation, next is filtration, precursor fading, immobilization, addition of cellulase, micropropagation, genetic transformation. So, what would be the answer for this question? A, C, D, G. Okay, A is elicitation, C is precursor fading, D is immobilization, and G is micro elicitation, precursor fading, immobilization. Okay, or Tejas, do you think that genetic transformation doesn't count? You cannot do any genetic transformation in your plant cells so that they cannot accumulate any secondary metabolites? The answer for this question is A, C, D, and G. Elicitation, precursor feeding, immobilization, and genetic transformation. So, what do you mean by elicitation? What do you mean by elicitation? Stimulate defense. You will add what? You will add elicitors. So you are trying to add elicitors so that they will go into their what mode? Stress mode and produce defense, try to do defense and then secrete secondary metabolites. So at which time point you will be adding elicitor? Like at which phase? Lag phase, log phase, stationary phase or death phase where you will add elicitor to your media. Any idea? Just a wild guess. Okay, stationary. So, but in stationary, yeah, maybe initially in the what you can call at the end of log phase and before the stationary even starts towards the end, before the log ends. Stationary phase is your right answer. So what does precursor feeding means then? What is the difference between elicitor and precursor then? What is precursor? The answer is in the word. What is precursor? So nobody other than Shashank has gone through the lectures. What is precursors? What are precursors? No idea. Say for example, um, you are having you are drinking coffee or tea. So coffee or tea has what in it? Intermediates and few pathway of product formation. Yes, intermediates. They can be intermediates or starting point of a pathway also. So a precursor is something which, say for example, if your end product is caffeic acid or caffeine, your starting point will be having some, especially from different pathways, right? You would have come across different pathways of secondary metabolic production. What else I can think about? So chlorogenic acid. There is a acid like it is a called chlorogenic. Say for example, chlorogenic acid is there. So in your pathway, um, chlorogenic acid is there, and after this also there are so many different end products will be there. So my area of interest is this chlorogenic acid, and before this there are various steps also. So one of the step is caffeic acid. After that, there are so many reactions. At one point, chlorogenic acid is being produced from caffeic acid. So, in this pathway, let us call it as a pathway, even though there are so many missing, what you will call as a precursor for chlorogenic acid. So, say for an end product, some end product is that we don't want this end product. I want only chlorogenic acid, and maybe the pathway starts from say X compound, and some there are plenty of other intermediates, and caffeic acid is also there, which I know the name. So there are still furthermore intermediates also present. So this is generally produced in abundance. So what you will do? What is precursor for what you can consider as a precursor for chlorogenic acid? Here in this example which I have given you, caffeic acid or something. Yes, Shashank is right. Caffeic acid you can consider as precursor because unless this is formed, this cannot be formed. So it is a precursor which is uh, before the production of something, or you can call it as unless this person is not being this thing is not going to be synthesized, you cannot go, not even go to this subsequent steps, which further leads to chlorogenic acid. So the precursor for this chlorogenic acid production would be caffeic acid if this is the pathway given. So this is the meaning of your precursor. So how does immobilization helps in enhancing the accumulation of secondary metabolites? How does you think so? Because you are going to encapsulate or entrap your cells inside a matrix. Let us consider this as a matrix. This is matrix. Your cells are inside. So you guys answered that immobilization is going to enhance your secondary metabolite accumulation in plant tissue culture. So how does it does? How does it do? Immobilization, how does it enhance your secondary metabolite accumulation? Reduces difficulties in DSP. Okay. How exactly it is reducing DSP? Because plant cells already tend to form aggregates, and you are now encapsulating them into an like capsule you're doing. So isolating from your liquid environment. There is one more mass transfer resistance or barrier being introduced in. So how do you think that reduces difficulties in this the downstream processing will come into picture here? Yes, your answer makes sense, but how you can explain it here? Any idea? Okay, this also we will discuss in the next class when you talk more about immobilization. So, genetic transformation, according to your answers, you guys have answered that genetic transformation will enhance the accumulation of secondary metabolites, and how does it so? What you can do with genetic transformation? How you will start a genetic transformation into plants and systems? Any idea? Just you can guess any answers, or you need time to think. Also, take 1 minute or 2 minutes. So, how do you think genetic transformation comes into picture in order to enhance the accumulation of secondary metabolites? Okay, forget about plants. How does generally genetic transformation done in microorganisms to improve any product? Like say, for example, improving synthesis of what what particular important products are there? We will take this example of chlorogenic acid or ethanol or something. How does you do genetic transformation which help in improving or enhancing the production of something? Bacterial mediated transformation by a particular organ where secondary metabolites are produced. Okay. Yes, bacterial mediated transfer. Yes, there are various transformation methods available. One of it is agrobacterium mediated transformation, agrobacterium tumefaciens or agrobacterium lysozymes you can use, or you can go, yes, gene cloning, correct. So either ways you can do, where you can transform particular gene of interest. Say for example, if we want uh, a compound, taxol or quinone or something like that, which has medicinal importance, and your plant is known to produce it, but it is producing way less than the needs of the market or than your anticipated need. So what you can do, you can clone the genes, you can clone the gene necessary for the synthesis. You can do metabolic engineering where you can down regulate or up regulate some genes and try to transform your plant cell systems into a higher producing strain, select, screen and select those strains, and then you can cultivate them so that it will have an enhanced production of secondary metabolites compared to your wild type strain. That you can do. So that is one way of increasing your secondary metabolic production or enhancing the accumulation of secondary metabolites in plant tissue culture. So we are discussing more about plant tissue culture and uh, why plant tissue culture and plant cell bioprocessing and all. So, so why do we even have to go for plant tissue culture? Uh, why we have to go for plant tissue culture? Any answers for this question? from the last 4 weeks, 3 weeks, we are talking about plant tissue culture, plants, bioprocess, plants, tissue, cells, those things and all. So why do you guys think that we go for plant tissue culture? Why can't we go for natural plant production? Like from nature itself, we can get it. Why we are going for plant tissue culture? What what are the reasons? Do you guys think that that is the these are the reasons? That's why we are going for plant tissue culture. Okay, so Shank has given an answer. High value product extraction. Okay, so um, how does high value product come into picture here? So same plants you can take it from nature and do high value product extraction also. No? Why you have to grow it here and then do the high value product extraction? Few reasons you guys tell me. So why we can't go for plant natural plant? Okay, one is good. Amount produced naturally is very less. Yes, the quantity of the particular see the even natural plants may be more, but the amount or quantity they produce is less. The quantity of product of your interest will be less. That is one answer. What can be the other answers? We cannot always find your plant of interest um, at abundance. Like maybe they are growing very slow due to climatic conditions, or they only grow. They are always slow growing plants only. There are various limitations to supply. Okay. So the other answer is for higher yield. Yes, for higher yield because like the answer which Sasank told is approximately here only. So endemic and endangered species, right? You are right. So the plant of your interest might be an endemic species or an endangered species, and you don't have necessary permissions to use them. Also, if they are under that endangerment and endemic criteria, you cannot use them for production because they are already on the verge or already endangered. So that is there. So what else? There will be batch to batch variations due to different factors like various environmental factors. Say for example, you want to collect what is the common plant? Aloe vera. Say for example. So aloe vera is commonly available, but you want to make some cosmetic something out from the aloe vera and you are cultivating it in different areas. Kerala, you are cultivating, Tamil Nadu, you are cultivating, Uttar Pradesh, you are cultivating, everywhere you are cultivating. Do you think the quantity of or quality of the product, say for example, we will see it is as a, say for example, aloe vera gel, will it be equal everywhere? Because one batch you are getting it from Kerala, one batch from Uttar Pradesh, one batch from Andhra, one batch from Jammu, something like that, from different places and different soils, everything matters, right? So everything will have, each will have different set of um, metabolites present, more or less similar, but will have certain variations happening all the time. Not only based on regions, even between the plants also there will be variations. So there will be more batch to batch variations will be there. To avoid that also we can go for plant tissue culture. And like Sashank said, lower concentration will be produced and also the what you call so the supply will be limited. There will not be always throughout the year. If you need a production throughout the year, you need the raw material throughout the year, but that may not be available in many cases. So that is also one of the reason where we shift to plant tissue culture, where the advantages of plant tissue culture would be there will be controlled production or controlled supply of raw materials throughout year because you are cultivating them, the plant cells or plant in vitro plants or in vitro somatic embryos, everything inside the lab. So there will be a controlled production happening um, throughout the air. So there is aseptically you guys will be doing everything from initiation to production, everything. So and also the amount of or the area occupied for cultivating that much amount of plant which are yielding this much amount of product. Say for example, 1 ton of your neem leaf is going to give you um, few kgs of your particular product means you can do that in the lab systems itself, where you can grow more cells within a confined space, so your time and space is also reduced. Because when aiming for a tree species like neem, for a neem tree to go, how long it will take? It will take a minimum 2 to 3 years for them to grow into a bigger tree. So that and all will be coming into picture. So when you go for plant tissue culture, there will be reduction in time and space, controlled supply throughout the year of raw materials, and you can expect a higher growth and productivity. And you can also tweak your cell systems, like you can do cell or strain improvement likewise you do in microbial systems. The same is possible here in plant tissue culture also. So so those are related to plant tissue culture and its advantages. So coming back to the session of like coming back to this week session, we have three more questions to answer. So if you have any doubts regarding plant tissue culture and plant cell bioprocessing, please feel free to contact me. I will give my mail ID at the end of the session. And do you guys so far had faced any difficulties or doubts? Strain improvement means, okay, Fauzi, strain improvement means what I can tell. How do you do strain improvement in microbial strain improvement means, say for example, you have a which cell we can take? Ashwagandha cell. Ashwagandha, everybody know, right? Ashwagandha, Withania somnifera. It is a good medicinal
Yeah.
plant. Yes. So let us have you have a plant which is ashwagandha, and you are cultivating it in what land also, and also in vitro inside the lab, and you have cells also, like callus cell lines of ashwagandha also you have at your lab facility. So what happens, say for example, a compound isolated from Withania somnifera is withanolides. So say for example, from the externally growing plants, your withanolide is going to be around for 1 kg you are getting some 100 no, 10 grams withanolide is you are getting from land grown plants. And your in vitro grown plants, say for example, 1 kg you are able to get 100 or say 50 grams. Your cells, 1 kg of your cell culture is going to give you around, say, example I'm telling, this is not exact values, I don't know these values at all. So, say for
Okay.
example, what happened?
No, man, no, man.
Ah,
Nothing.
so, yeah, 70 grams. Say, these are examples. So, this let us keep it as 100 grams. So, what happens? Your external land grown plant is going to give you around let's keep this 70 to 80. So your external plant itself is giving around 70 to 80, but the amount of time, space, water, everything comes into picture when you calculate this. What market accessibility or market rate. So your cells are able to produce, in case of 1 kg, they are able to produce 70 grams. So what you are trying to do is, you are trying to improve, you will plan to improve your cells to make them produce more than this 70 to 80 grams produced from the land grown plants, so that If you do strain improvement, they will even produce 120 grams. I will tell you how the strain improvement is done. One which you told that Fauzia was talking about gene cloning. You can do gene cloning. You can improve their yields by metabolically engineering them and doing or introducing your gene of interest to them, which can enhance the production of that particular compound alone. You can do that. And the next is like um, those techniques which we were discussing about, precursors you can add. Say for example, for this retinoid, say there is some precursor, say for example WA we will keep. This WA is a precursor for your retinoid. Means we can add this extra into your media. So what happens if this is in excess? This will also be produced in excess, right? Have you gone through this feedback mechanisms and stuff where if you don't understand, I can tell this. If I am producing my compound starts with A, it is going to form into B and then it will form into D and then it will form into D. So this is my starting point or starting compound and this is my end product. I want only D. So these A, B, C, all these of them can be my precursors. So if C is formed, then only this D will be produced, which is my end product of my interest. So what I can do is like I can add additionally C in my cell culture or suspension culture or mostly suspension culture. If I add so, the plant is capable of producing this C from A, B, C, D like that. It will keep on producing. If I add extra C, what it will do? It will think that it has more C in its system. It will divert all this compound C into compound D, which is my end product. So I will get more D, which is my ultimate aim. So I can do precursor addition. So elicitor also, if you find the pathway and if you think that that will aid in um, your enhanced production of that particular compound of interest, which is D here, then elicitor also helps. So you can do strain improvement by improving your either tweaking with your strain itself. Strain here by means your cell line only. So either you can tweak the cell itself or you can tweak your media components to make your cell produce more particular compound of your interest. That's all. That is strain improvement means. I hope you understood. If not, you can ask. Okay. So let's go to question number 6. So the micro-persistent molecular patterns maps are recognized by specific receptors, which then induces massive phytoalexin production. Such a defense strategy is referred as what? A innate immunity and B is acquired immunity. What would be the answer for this question? I think we have discussed this similar answers when we were discussing about phytoalexins. I even gave the answer for this question. So what will be the answer for this question? Innate immunity, right? Innate immunity. The answer for this question is innate immunity. So let's go to the next question. Age of the inoculum does not affect secondary metabolic production. Is the answer true or false? False. Okay. What does age of the inoculum means? What does inoculum means? Explant. Okay, explant is what? Any part of the plant is explant. Any excised part of the plant is explant. Inoculum is what? Cells culture we can call. In bacterial and other microorganism you call na inoculum volume, inoculum size or inoculum age they call. So yes, initial biomass concentration exactly. So why does the age you think that matters? It affects the secondary metabolic production. In previous classes we were talking about batch kinetics and all. So if you remember that, you can answer. Why does age of the inoculum affect the secondary metabolic production? Age of the inoculum affects secondary metabolic production. What are the other things which affect the secondary metabolic production? Physical and physical parameters. Let's see. What are the physical parameters which affect the secondary metabolic production? You guys told age of the inoculum affects the production. If you have an appropriate age of inoculum, you will get a good production, secondary metabolic production. It means that only, na? So what and all affect secondary metabolic production? If you want your secondary metabolic production, what and all needs to be optimized or what and all needs to be looked into? No idea. Nobody remembers, huh? Yes, Shashank is right. Temperature, pH, what else? Temperature is there, pH is there, your light, which is light intensity. Then, what else? Your media components, which includes your nitrogen, or I can tell, yeah, nitrogen, carbon source, macro micronutrients, what else? Phytohormones, what else? That's all, huh? Your RPM if you are going for suspension culture. These are all some of the things which will affect the cell growth also, and it will subsequently affect your secondary metabolic production. If these are all the things which needs to be optimized if you want to enhance your secondary metabolic production. So temperature. If any one of you can recall, what is the range of temperature we can use for plant tissue culture? What is the temperature range which you have to consider for use in case of plant tissue culture? No idea. Forgot, huh? Okay, forget about the temperature. pH, how much is the pH? Relatively, you give me the answer. No answer. Temperature 25 plus or minus 2 degrees Celsius. pH generally ranges from 5 to 6 for plant cells. Yes, correct, Shashank, you're right. So pH is 5 to 6. How much is light? How much is RPM? These and all, it depends on the plant species. So RPM is generally how much? Yes, Fauzia, right, exactly right. Because 5 to 6, it varies for plant tissue, plant species, but generally 5.8 people take for as an ideal option. So 5 to 6 is also right. 5.8 as a exact one. Yes, 90 to 120 RPM, right, Shashank? You're right. So what will happen if we go beyond this 120 RPM? So what will happen if we go beyond 120 RPM for the plant cells? both in suspension and in reactor. So what will happen? Cell damage. Yes, right. So cells will get damaged and they did, they will be like stressed out and that is not suitable for rotating because they are very shear sensitive. So generally it is recommended to keep it between 90 to 120 rpm. Still you can optimize them according to the plant species you are working for. Okay, we have come to the last question. So vacuole is able to trap the ions due to its dash environment, acidic or basic? Acidic. So why generally the vacuoles are in plant system? Why the vacuoles are generally present? What is their purpose actually? Why why vacuoles are present? Turgidity, okay. Turgidity to maintain turgor pressure, okay. Storage, okay. Food storage or something else? Yes, Ashta Mishra. Okay. Toxins and secondary metabolites. Yes, you are absolutely right, Shashank. Toxins, vacuoles are known to store toxins and secondary metabolites, and also in some cases precursors. They will store in the form of precursors so that the cell won't be get affected by the toxin themselves. So, is there vacuole is the only storage organelle present in the plants, or there are some other storage organelles which are available in the plants? Plastids, okay. Your plastids, your trichomes or glandular hairs, these are also organelles which can store um, your ions, like, or they can also store your toxins and other systems. Generally, they are assigned for storage. So one last question or discussion we can keep. Do you guys? Give me some plant-based commercial plant tissue culture based, not plant-based tissue culture based commercial products which are available in market. I hope you guys have, yeah, you have that in your selection content of last week. So, do you guys remember the plant tissue culture based commercial products which are available in the market currently? Coca-Cola, cocoa cells, okay. Fertilizers, antho and beta cyanins, okay. Cocoa cells for the production of what? Chocolate. Antho and beta cyanins are pigments. Okay, we can tell pigments or flavoring agents, we can tell. Okay, what else? Cocoa cells are there. And what else is there? Few fertilizers are there, like fertilizers are there. What else? Food colors, yes. Pharmaceuticals. Any example of pharmaceutical, if you remember? Berberine, okay. Berberine, one example. What else? Cosmetics, yeah, fragrance. Fragrance, cosmetics is also there. Anything we are missing? Food and food additives also, right? For sweetening agents, flavoring agents, everything. Okay. So, so these are some of the um, different areas where plant tissue culture is being commercially used for production of various products. So I think everybody has gone through the lecture videos very carefully and remembered many things. So if you have any doubts further, you can, I will give my email ID down. So if you have any doubts further, please ping me at my email ID. And um, have your assignments are being already submitted? Week 1 is done way before. Week 2 is done already. Week 2 assignments are still in due.
Yes, ma'am. 3 has gone.
Oh, 3 also has gone. Okay, good,
Hmm.
good. Okay, people. So if you have any further doubts, please contact me via my email ID. And if anything is, if any doubts you had which is not being resolved in your discussion forum also, you can remind me here so I can contact people involved in that. So if you don't have any other doubts, you can just leave the meeting. Thank you for joining. Thank you for joining the sessions. If nobody has any doubt, you can leave the call. We have come to the end of the session. So let's meet next week, same time, to discuss about the questions on week 5, and we will have a discussion on some of the topics which I gave as assignments this week. Thank you for joining.